Good morning, everyone. Thank you for joining. Today I'll be presenting a 2015 paper published in the Clinical Journal of Sports Medicine. The study is titled Acute Effects of Vitamin D Supplementation on Muscle Strength in Judoka Athletes. But as we'll see, the implications extend to dancers more broadly. It's a randomized, placebo-controlled, double-blind trial that asks a very specific question. Can we see improvements in muscle function, not in months, but in a matter of days following vitamin D supplementation? To set the stage, we need to understand the problem. We've known for some time that athletes who train primarily indoors, like gymnasts, dancers, and in this case, judokas, are at markedly higher risk for vitamin D insufficiency. This is simply due to a lack of sun exposure, our primary source for the vitamin. The link between vitamin D and muscle health is also well established. It operates through vitamin D receptor in the muscle tissue, and deficiency has been shown to cause atrophy, particularly in the powerful type 2 or fast-twitch muscle fibers. However, if we look at the literature, most of the research is focused on long-term supplementation over months, often in older or clinical populations. What's been missing are robust randomized control trials examining the acute effects, the immediate response in elite young healthy athletes. The question driving this study was simple. Can a single high dose of vitamin D3 lead to measurable improvements in muscle strength in just 1 week? To answer this, we used a rigorous double-blind, placebo-controlled design. The participants were 22 national-level male white indoor athletes engaged in 4 to 6 hours of daily training. The exclusion criteria are important to note. We excluded anyone who had taken vitamin supplements, traveled to a sunny climate, or had a significant injury in the past 3 months. This was to ensure that any changes we saw were truly due to the intervention and not external factors. The intervention itself was a single high-dose bolus of 150,000 international units of vitamin D3, administered immediately after the first test session. The placebo group received identical inert tablets. Critically, the timeline was very short. Participants were tested twice, once on day 1 at the baseline, and then again just 8 days later. This is the acute time frame the study is designed to investigate. So what was measured on day 1 and day 8? First, the primary biological marker, serum 25-hydroxyvitamin D, taken from the venous blood samples. Second, and most importantly, muscle strength. This wasn't just a simple grip test. We used isokinetic dynamometry, which is considered a gold standard for assessing muscle function. We measured the concentric peak torque of the quads and the hams on the right leg. We tested at 2 different angular velocities, a slow speed at 30 degrees per second, which is more indicative of maximum strength, and a fast speed of 200 degrees per second, which reflects power and recruits more type 2 muscle fibers. This was a clever design choice, as it might help reveal if vitamin D has a fiber type specific effect. A familiarization session and randomized testing order were used to ensure that data were reliable and free from learning effects. Let's look at the results. Starting with the blood work, the key finding here is that the vitamin intervention worked. The group that received the vitamin D bolus showed a significant 34% increase in their serum levels in just 8 days. The placebo group, as expected, remained flat. Statistical analysis confirmed a highly significant interaction between treatment and time. Interestingly, the researchers also performed a regression to the mean analysis. Without diving too deep into the statistics, this shows that the participants who started with the lowest vitamin D levels were the ones who benefited the most from supplementation. This is a clinically intuitive finding. Those with the greatest deficit had the most to gain. However, there's a crucial caveat here. Despite this 34% increase, the intervention group's post-test levels were still in the clinically insufficient range. They didn't reach optimal levels, which suggests that a single bolus, while effective, may not be enough to fully correct a deficiency. Now for the most striking finding. The increase in vitamin D was accompanied by a rapid and substantial improvement in muscle strength. The intervention group showed an overall mean increase of 13% in peak torque across all muscle groups and speeds. In contrast, the placebo group improved by only about 3%. The critical time by treatment interaction was significant, meaning the change was directly attributable to the supplement. Importantly, the improvements were seen at both the slow and fast speeds. This is interesting because it doesn't clearly support the idea that the effect is limited to just one fiber type. There was also a suggestion that the quads might have responded slightly more than the hamstrings. The authors are appropriately cautious about this, noting there's no strong theoretical basis for that difference. For an elite athlete, a 13% gain in strength in 1 week is a remarkably meaningful enhancement. So how do we explain a 13% increase in strength in just 1 week? It's highly unlikely to be due to the classic slow genomic pathway where vitamin D activates DNA transcription to build new proteins. That process takes weeks. Instead, we propose a more plausible, faster mechanism. There's evidence for a non-genomic pathway where vitamin D binds to a receptor on the muscle cell membrane. This can rapidly activate intercellular signaling pathways, influencing things like calcium transport and handling within the muscle cell. This could lead to an immediate improvement in the muscle's ability to contract forcefully. This finding is clinically significant. It suggests that for an athlete identified as deficient, we don't necessarily need to wait months to see a benefit. Optimizing their status in the week leading up to a key competition could have a real impact on performance. The study also highlights a potential difference between athletes and the general population. Some researchers, like Henley and Hollock, have suggested athletes may need much higher serum levels in the range of 120 to 225 nanomoles per litre to fully activate this muscle response, a level these athletes didn't reach. In conclusion, this study provides compelling evidence that a single high-dose bolus of vitamin D3 can lead to significant increases in both serum levels and critically muscle strength in deficient elite indoor athletes within just 8 days. So what are the practical takeaways for us scientists and practitioners? First, fundamentally, monitor your athletes. We can't assume their vitamin D status is optimal, especially in winter. Testing is straightforward and informative. Second, the response can be rapid. This opens up the possibility for acute interventions. If you identify a deficiency in the lead up to a competition, a high dose could provide a meaningful performance edge within a week. Third, we need to think about optimal targets. The levels considered sufficient for the general population may not be adequate for maximizing muscle function in elite athletes. This is an area needing more research. Of course, the study has limitations. The sample size is small and the treatment group started with significantly lower vitamin D levels, which, while analytically addressed, is a limitation. We also don't know what happened in weeks 2, 3, and 4. Serial measurement would give a much clearer picture. This study opens the door for further research into optimal dosing strategies and the precise mechanism behind these rapid acute effects. Thank you for listening. Please follow the DOI link to access the full article. And there's one more presentation in the vitamin D series.